Hey everybody, thanks for joining us today. Before we get to our speaker presentation, I want to give a very important announcement that we're excited to share real quick. So my name is Garrett Genther. I am with the Cell Analysis Division of Agilent Technologies, and today we wanted to share with you guys our newest development in flow cytometry, the NovoCyte Penteon flow cytometer. So just a brief background, if you guys haven't been familiar with Agilent flow cytometers, basically it was the first cytometer was launched in 2014 under ACEA Biosciences, and over the course of the next few years, we've been able to offer additional capabilities and also additional technology advancements to flow cytometry. So this includes the NovoCyte Quanteon and the NovoCyte Advanteon that have been launched in 2018 and 2019 respectively, and then today I'm announcing the newest cytometer to the family, the NovoCyte Penteon. So the NovoCyte Penteon is a 5-laser, 30-channel fluorescent cytometer building upon the technology built in the with the NovoCyte Quanteon. And when we look at the other cytometers available on the market in the 4-laser or 5-laser space, the Quanteon and the Penteon offer more channels, which equates to more flexibility for you. And our cytometers are upgradable. So Quanteon, a 4-laser NovoCyte Quanteon, can easily be upgradable to a 5-laser NovoCyte Penteon system, still using the advanced technology. So what is that advanced technology? If some of you guys haven't been involved or heard about the NovoCyte Quanteon before, traditional technology for detectors in flow cytometers has been using PMTs or photomultiplier tubes. With some of this advanced technology, we can achieve much more sensitivity in the detection of these fluorescence, the emitted fluorescence light. One of these detectors is called a silicon photomultiplier. It's a solid state photon level sensitive device, and it's really, it's well known for its high quantum efficiency. You can have high gain, and it's very robust. Even, so these are produced by Hamamatsu, and even they are very excited about them. It's on the their landing page on the website. So it's a really good technology. We've been using it for a couple of years now, and we've seen very, very good results in terms of sensitivity using these detectors. And so when we look at the UV channels that are now with the NovoCyte Penteon, so the Penteon will include a UV laser, a violet laser, blue, yellow, and red laser. The performance that we're seeing out of the UV laser with these advanced detectors is really nice. So at the top here, I just have, this is a quick plots showing you 6 peak beads and the separation there. But we also did some comparison with other instruments out on the market using the actual fluorochromes conjugated to CD4 and then bound to compensation beads. So in these channels, we were able to run these samples and with these various fluorochromes listed, and then calculate the stain index. So on the middle column is the stain index from the NovoCyte Penteon, and on the right-hand column is a stain index from a competitor instrument. And you can see with some of these brilliant UV dyes, we can see a 3-fold increase, like with BUV 395, or a 2-fold increase in the stain index, like with BUV 496 and 661. Now, we couldn't provide a comparison to all the channels because not all instruments offer as many channels as the NovoCyte Penteon. Now, sensitivity is really important in terms of being able to detect these dim signals that you may have on your cells, but also fluidic stability is really important in terms of reproducibility and delivering consistent results. So when we look at the fluidic design of the NovoCyte's line of flow cytometers compared to our competitor instruments, we see that no matter if you're running your sample speed at slow or fast, we still see a really, really tight CV. So CV is going to be the measurement of the variation in the signal detection, and when you have a higher CV, you tend to get more spread in your signal. With the NovoCyte Penteon on the left-hand plot, you see even at low speed, medium speed, and fast speed, there's a marginal increase in the CV, which would be expected, but it's very little. We're still seeing CVs below 5%, even at the high sample speed flow rate. Whereas in the competitor instrument, when you go from low to medium to fast, you actually see a rapid increase in the CVs as high as 15, 20, 25%, which is really going to produce more spread in your samples and the specific signal detection. Included with all of our NovoCyte line of flow cytometers is the very, very intuitive and powerful NovoExpress software. This is acquisition and data analysis software to provide you with as many automatic features as possible, like automatic report generation. We have a cell cycle analysis module, cell proliferation module, heat map display, all of these really to make the data acquisition and analysis as easy as possible. And automating these systems is very easy. We can either use an autosampler or we can attach this flow cytometer to a robot itself. So the autosampler can accommodate 42 racks, 96 well plates, 384 well plates. It has an integrated barcode reader, and it really provides walk-away functionality. So you can set a plate on the system, push run, and walk away, and be confident that you'll get good results when you return. And if you want to increase your throughput even more, you can connect it to a robotic platform. So I just want to show you some quick preliminary data that we've been able to generate using the NovoCyte Penteon. This was a 24-color panel analyzing whole blood and looking at different immune cell subtypes. Here's a list of all the markers. I don't have enough time today to go through all of them, but I just want to highlight a few of them to you. So as you can imagine, a 24-color panel can get really, really complicated and a little bit hard to display all the plots at once in one slide. So I'm going to focus in on some of these T-cell differentiation markers that we've been able to look at. So when we zoom in on these, up in the on the left-hand side, we have our gating strategy for identifying the lymphocyte population, separating those into T and B cells. And then in the upper right plot, we're looking at the gamma delta T cells. And this one is using one of the brilliant UV markers on the x-axis. And you can see that there's really good separation between the positive and the negative samples. On the right, the shaded blue and green areas, we're looking at different T-cell differentiation markers in the CD8 and CD4 cell populations, looking at central memory, naive, effector memory, and effector memory RA positive, CD45 RA positive cells. Up at the top, where the CD8 subset, and right underneath it is the CD4 subset. And you can see that these profiles differ pretty significantly between these two different types of T cells. And one other population I want to point out in the lower right-hand corner, you see that there's we're looking at the Tregs. So these are defined by CD25 positive cells that are also CD127 low. And you can see this population isolated here in the lower right-hand corner. So as you can imagine, this 24 color analysis can get really, really complicated, but you can use some advanced analysis features in order to tease out some of the differences in these immune cell subsets, like Tisney or Spade and whatnot. So with that, I just want to I just want to thank you for your time, and I want to now introduce Dr. Lara Campana. She has received her Ph.D. in Cellular and Molecular Immunology, focusing on the role of macrophages in tumor and autoimmune diseases. She is currently at the University of Edinburgh, dividing her time between her passion for teaching as well as her passion for understanding macrophage functions in various disease states. So today, I would like to welcome Dr. Campana.
Hello, everyone, and thanks for joining us. First of all, I would like to thank the organizer for giving us the opportunity to share our data here today. The project I'm going to introduce you today relates to the study of the role of STAT3 phosphorylation in macrophage phagocytosis as a potential therapeutic target in acute liver injury. We first introduce briefly what macrophage phagocytosis is and why it could be a potential therapeutic target in acute liver injury. I will then move to show you some results about the pro-phagocytic role of phospho-STAT3 in macrophages and how phospho-STAT3 controls autophagy and prevents immuno-senescence in macrophages. I will then move to show you and discuss with you our working model, and there will be then time for the Q&A part. Phagocytosis is all but a new topic. It was discovered over 100 years ago by Elijah Mechnikov, but despite Elijah Mechnikov already defined it as the one constant element in immunity, whether innate or acquired, there is still lots that we don't know about phagocytosis. Particularly, we don't know what happens to the macrophage after it has ingested its phagocytic cargo. In this presentation, I will use the word efferocytosis to refer specifically to the phagocytosis of dead or dying cells by a live macrophage. And I will define macrophages as professional phagocytes, as they are cells that perform phagocytosis and efferocytosis with high efficiency. Studying phagocytosis is crucial. In fact, inefficient phagocytosis is associated with recurrent infections, as you can see here on the right-hand side of your screen. Patients with COPD, chronic obstructive pulmonary disease, have recurrent infection, and their pulmonary macrophages are less able to phagocytose than those of healthy patients. Also, autoimmune diseases can develop as a result of inefficient phagocytosis. But what is exactly phagocytosis, and particularly efferocytosis, the phagocytosis of dead or dying cells? Phagocytosis starts with the sensing of the dead or dying cells, here indicated by a pink star. The macrophage is attracted to the dead or dying cells by find me signals, which are soluble molecules secreted or released by the dead cell. Here on the bottom right-hand side of the micrograph, you can see a macrophage that is protruding its membrane around the dead or dying cell. This is the phase of engulfing, when the dead cell exposes on its surface the so-called eat-me signal, cell surface molecules that are engaged by receptor on the macrophage cell membrane, forming the so-called apoptotic synapse. The macrophage then moves to internalize the cell, as you can see here. We know much less about this phase of phagocytosis than what we know about the previous steps of phagocytosis. In fact, there are no the so-called internalized me signal. What we know, though, is that for this phase to happen, the phagosome has to form and progressively be acidified. And this is key because we can move from the phase of internalization to the phase of digestion of the phagocytosed dead cell, when the phagosome fuses with the lysosome and the phagocytosed cells is dismembered. But what has it all to do with acute liver injury, and why are we interested in acute liver injury? We define acute liver injury as a single hit to a liver that causes a damage. And by far and wide, both in the US and the UK, the single major cause of hospitalization following acute liver injury is acetaminophen, also known as paracetamol overdose. When acetaminophen, or APAP for short, is ingested at therapeutic doses, it is normally very safely metabolized by the liver and is then excreted by via bile or urine. However, if the APAP is ingested in a toxic dose, in an overdose, it is metabolized in the liver by the CYP2E1 enzyme, a member of the cytochrome P450 family, and converted into this intermediate molecule called NAPQI. NAPQI can rapidly conjugate to glutathione, and if in excess, it can rapidly deplete all the glutathione pool of a cell, inducing oxidative stress. Furthermore, NAPQI can conjugate with other cellular macromolecules, for example, phospholipids, and the two pathways converge into inducing the hepatocyte cell death by necrosis. In this hematoxylinase staining, you can see indicated by CV the central vein of the liver, surrounded by an area of necrosis that is indicated by the black arrow. And you can clearly see the striking difference by the healthy parenchyma indicated here by the P and the necrotic areas, where hardly any healthy hepatocyte can be found. And this can give you a hint why macrophages, and particularly macrophage phagocytosis, can be crucial in this setting. In the case of a liver damage of any kind, the hepatocytes in the liver die, and an inflammation follows. The first cells to come to the liver are the neutrophils that die in situ once they're spent. The second wave of inflammatory cells coming into the liver is represented by macrophages that are able to scavenge both dead hepatocytes and dead neutrophil. This efferocytosis is instrumental for the conversion of the macrophage phenotype from pro-inflammatory to pro-restorative and to liver regeneration. However, when an overdose of APAP occurs, the liver damage is extensive and the number of that hepatocyte increases. And this creates a very extensive inflammatory reaction with an increased number of neutrophils that infiltrate the liver. This means an extra work for the macrophage that have to scavenge more dead hepatocytes and more dead neutrophils. A particular phenomenon that we observe in the liver of APAP overdosed mice is that the resident cells, the resident macrophages of the liver, also known as Kupffer cells, are depleted. And instead, they're replaced by a wave of infiltrating macrophages that are monocyte-derived macrophages. This latter population, though, is much less efficient at performing phagocytosis than the resident macrophages. So basically, during APAP overdose, we find ourselves, for reasons that are still poorly understood, in a situation in which we have lots of macrophages that are not able to perform phagocytosis efficiently, and this leads to a lower percentage of macrophages converting their phenotype into pro-restorative macrophages and decrease liver regeneration. In order to study macrophages in acute liver injury, we make use of this particular mouse model. This is a mouse that lacks the expression of the molecule GPNMB. GPNMB is a one transmembrane protein that is instrumental for the maturation of the phagosome. Therefore, macrophages lacking GPNMB can internalize the phagocytic cargo, like the macrophage on the left-hand side of your screen, but they cannot digest it, so they cannot become like the macrophage on the right-hand side of your screen. These mice lack the expression of GPNMB because a stop codon, that is the result of a spontaneous mutation, is introduced in place of an arginine in position 150, which is very close to the end terminal of this protein. Therefore, the protein is non-functional and the phagosome cannot mature. If we treat the GPNMB proficient versus GPNMB deficient mice with a mild to moderate overdose of APAP of 300 mg per kg, and we then harvest the livers at various time points after the overdose, we are able to compare the necrosis extent in the two strains of mice. As you can see, at 8 hours, there is a trend towards having a bit more necrosis in the GPNMB deficient mice. However, this difference is not significant, and the difference is also not significant at 10 and 36 hours. However, at 60 hours, when the GPNMB proficient macrophages mice have completely healed their liver, as you can see from the hematoxylin-eosin staining below. The GPNMB deficient mice that have macrophages unable to digest the phagocytic cargo still have extensive necrosis. Extensive necrosis can be the result of lower hepatocyte proliferation, higher hepatocyte cell death, or reduced macrophage phagocytosis and wound healing functions. Our studies seem to indicate that proliferation of hepatocytes is not the matter here, and hepatocytes do not seem to die more to start with. As you can see, at 10 and 36 hours, as we said, the necrosis is absolutely comparable in the two groups of mice. Therefore, we are leaning to think that whenever macrophages are inefficient for phagocytosis, even a mild to moderate overdose of paracetamol can result in a failed liver regeneration. Therefore, our working hypothesis is that it is crucial to understand the molecular pathways that keep phagocytosis efficient, and this is instrumental to identify potential therapeutic targets to improve the outcome in patients with acute liver injury. In 2018, we published a paper in which we showed that one of those pro-phagocytic pathways is the phosphorylation of STAT3. Following the phosphorylation of STAT3 in inflammatory macrophages performing efferocytosis, the macrophages are able to convert their phenotype to a restorative phenotype. Also, the correct expression of STAT3 phosphorylation is crucial to time hepatocyte proliferation, so that hepatocytes can proliferate only after the liver has been cleared from all the necrotic tissue. In order to perform this study, we decided not to use Western blot, because with a Western blot you can only analyze the bulk of cells that perform phagocytosis, but you cannot distinguish in your bulk the macrophage that have from the macrophage that have not performed phagocytosis. Therefore, we decided to use a flow cytometry approach in which we can label the phagocytic meal with a fluorescent dye, and we can therefore recognize specifically which macrophages are performing phagocytosis and interrogate only this subpopulation for the expression of phosphorylated STAT3. In order to analyze multiple time points and genotypes or treatments at the same time, we also developed a medium throughput screening flow cytometry assay that I'm going to describe to you in a minute. The base for this assay is a simple flow cytometry assay in which macrophages are labeled with any macrophage cell surface marker. We use, by way of example here, CD11B. And the meal is, in this case, is apoptotic thymocytes, is labeled with a dye, which in this case is TMTMR. And the double positive cells are those macrophages that have internalized the phagocytic cargo. With this assay, we're able to analyze multiple time points and multiple phagocytosis to target ratio ranging 1:2 to 1:20. Unless otherwise stated, all the experiments you will see here are performed with a phagocyte to target ratio of 1:5. On the right-hand side of your screen, you can see the result of our first experiment done with this assay. And in this assay, we have measured the phagocytosis by GPNMB proficient and GPNMB deficient bone marrow-derived macrophages. And as you can see, the proficient macrophages that are proficient for phagocytic cargo are increasingly able to phagocytose over time, while the GPNMB deficient mice that are deficient for the phagocytic cargo are not. We then moved this assay from the single tubes into a 96-well plate. We mixed the bone marrow-derived macrophages and the apoptotic thymocytes labeled with CMTMR in a ratio 1:5, and we analyzed phagocytosis and STAT3 phosphorylation at distinct time points. Briefly, what we do is that we label the cells at the end of phagocytosis, we fix and permeabilize them, and we perform the intracellular staining. We then wash the cells from any excess staining, and we read them using the NovoCyte 3000, and we analyze the data using the NovoExpress software. As a negative control, we use fed bone marrow-derived macrophages treated with cytochalasin D, and also unfed bone marrow-derived macrophages. The first experiment we performed was with this platform was a time course using bone marrow-derived wild-type macrophages fed with apoptotic cell in a ratio 1:5. And we analyzed the phosphorylation of STAT3 at 15, 30, 60, and 120 minutes after the start of phagocytosis. And we performed the analysis only in the gate of phagocytosing macrophages. And as you can see, STAT3 is phosphorylated early on during phagocytosis. The phosphorylation is maintained at 30 and 60 minutes before declining around the 120-minute time point. We then moved to compare the GPNMB proficient and deficient mice to see whether the GPNMB deficient mice were less able to phosphorylate STAT3 upon ingestion of the phagocytic cargo. Interestingly, both the GPNMB proficient and deficient mice are initially able to phosphorylate STAT3, as you can see here, 7 and 15 minutes. However, the GPNMB deficient mice have a progressive and faster decline in the phosphorylation of STAT3, probably as a result of their lower ability to digest and process the phagocytic cargo. And therefore, from this experiment, we could conclude that cargo digestion partially controls phosphorylation of STAT3 during efferocytosis. We then moved to analyze whether this lower activation of STAT3 had any functional meaning. And in order to do so, we performed an assay in which we took wild-type bone marrow-derived macrophages and we treated them with a pharmacological inhibitor of STAT3 to verify whether this induced any functional defect in the macrophages. We used two different approaches. One, for the first experiment, we used live imaging, and then we confirmed the result using the phagocytosis assay I've just described to you. And as you can see here, cells that are macrophages that are treated with a phospho-STAT3 inhibitor are less able to perform phagocytosis as compared with the vehicle-treated counterparts. Interestingly, if IL-6 is added to the cell culture as a mean to exogenously stimulate STAT3, the phenotype is partially rescued. At the time of the publication of this paper, however, we couldn't quite make up why phospho-STAT3 had these pro-phagocytic functions in macrophages. So moving forward in the project, we focused on what is a macrophage unique challenge among all other cells in our body. Macrophages have to recycle a sheer amount of other cells' waste when they efferocytose them. And one pathway that is ubiquitously used by all cells in our organs and tissue to cope with cell stress is autophagy. Autophagy is a way in which the cells, when are in need, in an energetic deficit, can recycle their own organelles and therefore can survive a certain stress. Macrophages are probably not different from any other cell. So if they are in the steady state as something perturbs them, they're able to induce autophagy. Therefore, we started to hypothesize that probably what phospho-STAT3 does is that it induces a short boost of autophagy in the phagocytosing macrophage, and this allows the macrophage to become or to maintain its phagocytosis efficiency. The link between autophagy and phagocytosis is by no means new. There are several papers out there pointing out at a relationship between activation of autophagy and efficiency of the phagocytic process. However, the mechanism that link the activation of autophagy and the maintenance of phagocytosis efficiency is still poorly understood. And we hypothesized that the phospho-STAT3 and cargo digestion are indeed linked to the activation of autophagy. In order to prove this hypothesis, we took bone marrow-derived macrophages from GPNMB proficient and deficient mice. We sorted, using FACS sorting, the phagocytosing GPNMB proficient or GPNMB deficient mice, we extracted RNA and analyzed the expression of a number of autophagy-related genes. And as you can see, in GPNMB deficient mice, macrophages, which I remember you are deficient for cargo digestion, a number of autophagy genes are less expressed, including AMBRA1 and ALK1, which are exquisitely autophagy-specific genes. We also performed an electron microscopy analysis, and we counted the number of early autovesicles and late autovesicle per macrophage. The yellow arrow indicates what an early autovesicle looks like, and the red arrows indicate what a late autovesicle looks like. And we quantified the early or late autovesicles in macrophages that didn't perform phagocytosis or that are in early or late phagocytosis. And as you can see from the central panel, macrophages that are in early phagocytosis, which basically are macrophages like the one you see in the micrographs reported here, they have a lower number of late autovesicles. So this convinced us that macrophages activate autophagy following the digestion of their phagocytic cargo. Now, if this is phospho-STAT3 dependent, we should see a similar phenotype in wild-type macrophages that we treated with an inhibitor of phospho-STAT3. As you can see here, from the quantification of the early and the late autophagosomes, bone marrow-derived wild-type macrophages that have been treated with a pharmacological inhibitor of STAT3 during phagocytosis indeed have a lower number of late autovesicle, mirroring what happened in the GPNMB deficient macrophages. Interestingly, if we add IL-6 to the cell culture as a means to stimulate exogenously the phosphorylation of STAT3, the number of late autovesicle is restored. And I remember you that IL-6 was also able to restore the function, the phagocytic function of these macrophages. We then discussed this data with this colleague of mine, who is an exceptionally talented postdoc, Sofia, who is very interested in studying cellular senescence. And she pointed out that my phospho-STAT3 inhibitor-treated macrophages very much showed pathognomic sign of senescence, which are those electron-dense accumulation that you can see here in the picture, indicated by the pink arrow. And I went back to my GPNMB positive and negative micrographs, and I could see that those accumulation of proteins were present also in the GPNMB deficient mice. And if we analyzed the RNA of the phagocytosing GPNMB proficient versus deficient mice, we could see that the single gene that is significantly upregulated during phagocytosis in the GPNMB deficient, so cargo digestion deficient macrophages, is CDKN2A, which is indeed a gene related to cellular senescence. We started then to get our mind around the concept of immuno-senescence, and we discovered that in literature, macrophage immuno-senescence is described as a condition that appears with aging, in which macrophages can show an increased pro-inflammatory profile and decrease phagocytic capacity. So we became convinced that perhaps what happens in condition of cargo overload is that the macrophage undergoes a premature aging, encountering immunosenescence. Now, if this was, again, a phospho-STAT3 dependent pathway, we should observe a similar upregulation of senescence-related genes in bone marrow-derived macrophages, wild-type bone marrow-derived macrophages treated with a pharmacological inhibitor of phospho-STAT3. And indeed, when we analyze the RNA of these macrophages, CCND1, which is another senescence-related gene was upregulated in the phospho-STAT3 inhibitor-treated macrophages. The addition of IL-10 as a mean to exogenously stimulate the STAT3 phosphorylation reverted the increased expression of CCND1. We also observe a trend for P21 another senescence-related gene, but we didn't see any regulation or up or down regulation of RB1, which is typically a apoptosis-related gene. So this really convinced us that in the absence of a correct STAT3 phosphorylation upon cargo digestion, macrophages undergo immunosenescence. To finally prove it, we needed to show that these macrophages indeed showed a pro-inflammatory phenotype. In order to do so, we dosed the number of cytokines and chemokines in the cell culture supernatant coming from these phagocytosing macrophages. In the column on the left, you can see the expression level of various cytokine and chemokines of macrophages that do not perform phagocytosis. As expected, the macrophages that perform phagocytosis correctly, that is the central bar in all these graphs, express lower level of pro-phagocytic molecules such as KCGRO, MCP1, or MIP2. However, when the same macrophages are treated with a pharmacological inhibitor of STAT3 phosphorylation, then the repression of the pro-phagocytic molecules is completely lost. And indeed, macrophages that are treated with a phospho-STAT3 inhibitor show an increased inflammatory profile. This further convinced us that this was really a pathway that was worth considering to target pharmacologically in order to improve phagocytosis during acute liver injury. However, as you may be aware, phospho-STAT3 controls a zillion of other functions, and therefore we need a better understanding of what is the network of genes that is activated by STAT3 phosphorylation, downstream STAT3 phosphorylation in phagocytosing macrophages. In order to do so, we used a proteomic approach that compared the proteomic profile of vehicle-treated, of phospho-STAT3 inhibitor-treated macrophages from 3 different mice. The aim was to compare the protein expression in the presence or in the absence of the inhibitor. Among all the proteins that were detected in this assay, 43% of them were expressed in all of the 3 preparations coming from the 3 different mice. And therefore, we focused our attention on those. Of those 1,278 proteins, 85 of them were up or down regulated in a significant manner in at least 2 out of 3 preparations, and 8 of them were up or down regulated in all of the 3 preparations. And what we did first was to use the STING pathway analysis tool, and we input all the 85 proteins that were up or down regulated in at least 2 out of the 3 samples. And as you can see here, most of them are actually related, which made us think that actually phospho-STAT3 can be the start of a very coordinated cellular response that gives rise to a number of the activation of a number of different pathways. We then focused further our attention only on the 8 hits that were up or down regulated in all the 3 preparations. And we perform some gene ontology analyses, and we could see that the pathway that were mostly regulated as a consequence of the STAT3 phosphorylation inhibition were pathway that had to do with epigenetic regulation of the SMAD pathway, and interestingly, regulation of the Wnt pathway. The Wnt pathway is relevant because previous Ph.D. students in the lab worked on the role of Wnt as a phagocytosis-related pathway during biliary disease, which is indeed another form of liver disease. We also analyzed some other parts of that big pathway that you could see before. And interestingly, a part of that pathway indeed relates to oxidative stress-induced senescence, which further and independently confirm that the inhibition of phospho-STAT3 during phagocytosis leads to macrophage developing immunosenescence. These are very preliminary data. This is a very recent experiment. It is by no means conclusive, but we hope that by analyzing it further, we can come up with some interesting targets to inhibit or stimulate with a small molecule in order to improve the outcome of acute liver injury. So far, our data prompted us to design this model in which, upon efferocytosis, STAT3 is phosphorylated in the macrophage. And the STAT3 phosphorylation is instrumental to the activation of autophagy, which prevents the development of immunosenescence and promotes the conversion of the macrophage from a pro-inflammatory to an anti-inflammatory pro-restorative macrophage, which in turn is has a positive effect on liver regeneration. However, if the cargo is not digested efficiently, STAT3 cannot be phosphorylated, autophagy cannot be activated or is activated in a suboptimal manner, and the conversion of the macrophages is equally not happening or suboptimal, and this has a negative impact on liver regeneration. At the end of this presentation, I would like to thank all those people without whom this project wouldn't have been possible in first place. First of all, my postdoc supervisor and mentor, Professor Stuart Forbes, all the people in the lab, particularly Rona, Sophia, Ben, and Janet, all our external collaborators, all the facilities, both at the Centre for Regenerative Medicine and in other centre of the University of Edinburgh, such as the King's Buildings and the CR UK Centre, all our funders. And I thank all of you for your attention, and I will now be happy to answer your questions.
All right. Thank you very much, Lara. Very interesting. I want to make one announcement that I forgot to make at the beginning, just while we're waiting for some questions to come in. If any of you guys are interested to get more information on the NovoCyte Penteon 5-laser flow cytometer, please go to agilent.com, and under products, you can click on flow cytometry and find the instrument there. I would give you the website, but it's way too long. So that is the easiest way to find more information. You'll see some of the information that I presented here earlier. So right off the bat, there's one technical question for you today, Lara. It's about your intracellular phospho-STAT3 stain. Are you able to, can you kind of explain how you were able to stain for phospho-STAT3 in some of the assay conditions?
Yeah, sure. With an antibody against phospho-STAT3, particularly against the tyrosine in position 705, which is the phosphorylation we are particularly interested in this context. And yeah, it's commercially sold by BioLegend, so we just buy it off the shelf, really. And then we perform our cell surface staining for CD11B as a marker, like a general myeloid marker, which is by no means specific for macrophages, but because we are using bone marrow-derived macrophages, is only macrophages that is there in our cell culture. We've previously shown that our cell culture is 99.8% macrophage pure, so we use CD11B quite confidently. We then fix and permeabilize the cell, and then we perform the phosphorylation of STAT3. At the start of this project, we had to perform also an immunofluorescence, which showed that, interestingly, there is both a nuclear and a cytoplasmic localization of STAT3 phosphorylation, and our flow protocol specifically detects the cytoplasmic one, is not a protocol for nuclear staining. I hope this answered the question.
Yes, it did. Thank you. I have another question in the context of autophagy and phagocytosis. So do you think this is more of an energy demand, or is it like a nutrient type of demand? And if so, if it's one of those, do you think that you could rescue it by exogenously supplementing it?
Yeah, that's really interesting. So my feeling is that it's a bit of both. This is because even like once I just literally made a mistake, and instead of feeding my cells at different time points, I fed them all together. So what I did, I tried to do sequential phagocytosis, in which the first phagocytosis didn't have phospho-STAT3, and the second phagocytosis did have phospho-STAT3. And despite the second phagocytosis happens in fresh medium, the phagocytosis is still deficient, even if I'm not blocking STAT3 directly, and even if the medium is fresh. So I think that there might be a component of, nutrients need, but I think really what matters more here is probably the cellular stress that is triggered by the ingestion of several dead cells at once. So I think it's more of a cellular response to stress. And one thing I've not mentioned, because it's also quite preliminary, but we think there is also a bit of mitochondrial dysfunction, because we see this very big ballooning mitochondria in the GPNMB deficient macrophages when we analyze them by electron microscopy. So I think it's more an energy deficiency, is more an energy problem rather than the nutrients.
Oh, very cool. Yeah, it would be very interesting to be able to supplement, to give those cells more energy, and then they could more efficiently perform the phagocytosis even without having the autophagic functions intact. So yeah,
and it's also interesting, I think, that the cells need to activate phagocytosis at a very specific time point. So something I'm not showed here today, but we're including in the paper, is that if we pre-induce autophagy by, for example, removing the serum from the medium before exposing the macrophages to the meal, the macrophages are still are again inefficient for phagocytosis. So it seems that it has to be really timed, and we think that these kind of timers can be phospho-STAT3, so the phosphorylation STAT3.
Very interesting. Very nice. Thank you. We have a couple of questions coming in regarding some of the flow cytometry assay, the 24-color panel that I mentioned in the very beginning. And just because that'll take up quite a bit of time and there's so many markers that were used, I will follow up with those questions over email. There's another question that has come in terms of senescence. How would you define senescence how do you define senescence in a post-mitotic cell, kind of like the macrophages?
Yeah, that's a very good question. So normally when we think of immunosenescence, we think of T cells that become unable to do clonal expansion, or less efficient at doing clonal expansion. Actually, it has been shown that macrophages in elderly patients are less efficient for phagocytosis and are more pro-inflammatory, a concept that has recently been crystallized with the term inflammaging. So the fact that as we progress in age, our body becomes a progressively more pro-inflammatory environment because of this lack of modulation that the phagocytosis brings on the innate immune system. And some studies have correlated this with some chronic conditions that are typically appearing with aging as well. So what we think it happens here is that even if you're in your 20s, but you take an overdose of paracetamol, the liver undergoes such a massive necrosis and intense oxidative stress that the macrophages become old, meaning that they become less efficient in performing phagocytosis, and they become more pro-inflammatory, thereby becoming part of the problem instead of part of the resolution of the problem. And that's why we think that's a very interesting target in the context of this disease.
Okay. Thank you. There's another question that came in, and I think you touched on it on one slide with some of the other publications. But do you also see where else in the body are we seeing this link between the autophagy requirement for efficient phagocytosis by the macrophages?
Sorry, can you repeat the question, please? Yeah.
So the question is: are there other organs or places in the body where phagocytosis is dependent on autophagy, or maybe other contexts in terms of disease or challenges or injury that you can elaborate on?
Yeah, phagocytosis has been shown to be instrumental, for example, to prevent bacterial colonization in lungs. So patients that have chronic obstructive pulmonary disease have less efficient macrophages in terms of phagocytosis, and they are indeed exposed to recurrent bacterial infections. I've not mentioned here for sake of time, but also in acute liver injury, one of the major problems in our patients is that they show an increased translocation of bacteria from the gut to the circulation, because the liver normally is a barrier for those bacteria, and because the macrophages are less efficient at performing phagocytosis, they can't perform this barrier function anymore. Not necessarily linked to autophagy, but phagocytosis, like a deficient phagocytosis, has been linked also to the pathogenesis of some autoinflammatory autoimmune diseases, such as systemic lupus erythematosus. And that's because that cell remain around and move from apoptosis to secondary necrosis, and they release their intracellular content, and this kind of perpetrates and then further elicits inflammation and then the following immunological response. So yeah, those are two other examples. I think someone made a very interesting comment here saying, don't forget that autophagy is also part of the reparative process in which cells have the opportunity and time to do what they need to do. And I totally agree with that. In fact, we think that the reason these autophagies kicks in during phagocytosis, that normally these would have been designed to give the macrophage the time to digest and deal with all the internalized cargo, and to then progress and eat more cells. So definitely we think this is how it has kind of physiologically evolved and what it has physiologically evolved for. We just think that it may be dysfunctional in the case of acute liver injury, just because of this combination of a very high level of cell death and a very oxidative environment.
Okay, yeah, that's a great point. Thank you. So, all right. Any other questions coming in from the audience? Let's see. All right, I think that's all we have for now. Please, let's thank Lara Campana. I really appreciate your talk. It was very interesting, and I hope you enjoy the rest of your day, everybody.
Thank you.